Hey guys, hope everyone's doing really well. Assalamu alaikum, this is Dr. Nerd, and today we'll be talking about the citric acid cycle, also known as the Krebs cycle. So aerobic respiration consists of three basic steps. Carbon from the glucose is converted to acetyl-CoA through the process of glycolysis and after going through the PDH complex. We're going to talk about both of these in a little bit more detail later on. So this carbon doesn't just come from carbohydrates that are broken down to glucose, it can also come from proteins, which is going to get broken down to amino acid and then to acetyl-CoA directly. There's no glucose intermediate in between or whatever. And it will also get converted, like fats can also get converted to glucose, which takes place in the liver. There's particular enzymes for that. And so this carbon can come from all three: carbohydrate, fats, and protein. This acetyl-CoA is then going to be oxidized in the Krebs cycle to produce the following products: carbon dioxide, which is a waste product and it is excreted in the lungs through exhalation, reduced electron carriers, and ATP as well. So these reduced electron carriers will then get reoxidized, which is step 3, and this happens in the electron transport chain later on, and this is going to produce a lot more ATP. This is basically where the most ATP is produced, through the electron transport chain. But that's a topic for another video, inshallah. Okay, glycolysis. This is the first step. The raw material is glucose here, so this is the process through which the one molecule of glucose is converted or broken down to form 2 molecules of pyruvate. And there are some small compounds that are formed or made as well. This process is completely anaerobic, it does not require any oxygen whatsoever, and it occurs in the cytoplasm. So basically this diagram particularly can look quite daunting, but I'm going to have you look at just a couple different things. So we can see there's an ATP molecule getting used up here, another ATP molecule getting used up here. So that's a deficit of 2 ATP molecules. Then we have 2 being produced here as well as 2 being produced at the bottom here. That's 4. So we're producing 4 molecules, but we're using up 2. So that is a net gain of 2 ATP molecules, which is why we say that it actually just produces 2 ATP molecules. This process of glycolysis also produces 2 NADH, which is an electron carrier, and this is going to directly go to the electron transport chain and get reoxidized. And other than that, there are 2 pyruvate molecules as well. So glycolysis ends up with the following products: 2 molecules of ATP, 2 of pyruvate, and 2 NADH. That's the end products of glycolysis, sorry. Pyruvate dehydrogenase. So now that we have pyruvate, we can't just simply put it into the Krebs cycle. It doesn't work that way. We first need to convert it into acetyl-CoA, and it is the acetyl-CoA which is the fuel for the citric acid cycle. So basically, PDH, or pyruvate dehydrogenase, is a multi-enzyme complex, which means that it is a complex which is made up of multiple enzymes. It's not just one enzyme that's going to work on pyruvate and then give you acetyl-CoA. So basically, this group of enzymes is made up of 3 basic ones. You have pyruvate decarboxylase, which is shortened to just E1 to make it easier to reference later on, dihydrolipoyl transacetylase, which is E2, and dihydrolipoyl dehydrogenase, which is E3. And together, E1, E2, and E3 make pyruvate dehydrogenase complex. These are found only in the mitochondrial matrix. So just a short summary, or just a short reminder on what mitochondrial matrix is. Basically, if this was your mitochondria and you have these inner cristae inside, the space inside would be the matrix. That is what the matrix is. And this is where you'd find the PDH complex. Basically, this complex is responsible for the oxidative decarboxylation of pyruvate, and this term is pretty self-explanatory. It just basically means that you're removing a carbon from pyruvate, and we'll see that in a little bit more detail in a second. This step is irreversible, and if I wanted to shorten this entire reaction into just one equation, I'd say that you have over here, you have pyruvate, NAD+, and CoA, and together the end products are acetyl-CoA, carbon dioxide, and NADH. So if we were to look at this diagram in a little bit more detail, look here, we can see that one molecule of pyruvate has 1, 2, and 3 carbon molecules. However, acetyl-CoA, to convert into acetyl-CoA, you only have 1, 2 carbons, which means that we need to get rid of 1 carbon, and this is what decarboxylation is. You're producing 1 molecule of carbon dioxide. This is where 1 carbon is kind of given off. So this carbon dioxide is obviously exhaled, it's excreted in the lungs, it's a waste product. So an NAD+ is reduced to NADH, right here. And you need this as well. This is a raw material as well, and that's the pyruvate dehydrogenase complex. Acetyl-CoA and NADH would kind of inhibit these, because acetyl-CoA and NADH are both produced when there is plenty of energy in the cell, and when you have plenty of energy, these high energy compounds would kind of build up, and when you already have a good amount of energy in the cell, you don't really need to produce anymore. So this kind of inhibits PDH, so you don't have acetyl-CoA, and it won't go into the Krebs cycle. You won't get any more ATP. This is just kind of a way of regulating the rate of the reaction. So just like limiting the amount of energy that's being produced if you already have plenty. Pyruvate dehydrogenase complex, it requires coenzymes. There's 5 coenzymes basically that are required. A coenzyme is basically a non-protein compound which is absolutely essential in the functioning of an enzyme, or in this case, a group of enzymes. So the 5 enzymes that are required are thiamine pyrophosphate, lipoic acid, CoA, FAD, and NAD+. So if you were to take the initials of all of these, T, L, C, F, and N, you can kind of come up with this mnemonic, which is the lovely cofactors, or coenzymes actually, for nerds. There you go. So this has the initial of all of the 5 coenzymes. This is just a mnemonic to make it easier for you to remember. Now, why is this particularly important is because these same 5 coenzymes are later going to be required for another enzyme, which is alpha-ketoglutarate dehydrogenase. We're going to be talking about that in the Krebs cycle itself.